So I'm going to talk about adaptive optics. There has been actually a wonderful demo also here at Me4Bio by Alexandra Fragola. I don't know if it's still on show, but maybe it's worth visiting it. It's really amazing. For those who haven't been at the workshop, the first half of my presentation will be going along the same lines. So I would like to present some introductory tour for people who are interested in the technology for microscopy. And the second half will be more about my current research in my team on this topic. So AO, probably it's very familiar that the technology comes from astronomy, and the proposal was done in the 1950s. Yeah, maybe we can actually counteract atmospheric turbulences with deformable elements fast enough such that the wavefronts coming from the stars are flattened out all the time, right? And I can see galaxies, planets, and stars crystal clear. Now in astronomy that was put into practice in the 90s, but of course in biological imaging we don't have an atmosphere we have to look through, but we have to look into samples, and that becomes more and more important actually. So this is actually should be a slideshow here. I don't know, it doesn't work. Never mind. But the left works, right? These are images we took with a 2-photon fluorescence microscope, just demonstrating that even with a tool that is actually dedicated for deep tissue imaging, is struggling to look inside these 200 micron thick cancer spheroid. Yeah, and the same you would have seen on the right. These are GFP expressing neurons in a human brain organoid. And after 200 micron, you see there is just no signal left. Yeah, it's extremely challenging to go inside these tissues, and this is where AO can help. Now, for biomedical imaging in ophthalmology, AO is, I think, most developed, largely because they share the same tools as the astronomers. They have direct wavefront sensors and kind of the same feedback loop. You can buy actually commercial AO systems for ophthalmology. Also for biological imaging, it's not really a new technique. So the thing started around the year 2000. And what you see here, I think, is one of the first confocal microscopes that has been equipped with AO. That was Martin Booth's group in Oxford, where he used the deformable mirror to get improvements in confocal images. So nowadays, there's lots of different tools available to actually do the wavefront shaping. That's actually not really a problem. You can buy those things. They're really good for different purposes. There are deformable membrane mirrors, segmented elements, liquid crystal light modulators. There's a bunch of optofluidic light modulators available now since a couple of years. And I've seen you can also do SLMs with thermal actuation. Yeah, it's a pretty new development. Yeah, very exciting actually. So they are here. But what are you going to do with them? Yeah, I want to say a few words about this. You have a microscope. You want to buy an SLM. That's the general term for it, spatial light modulator. What are you going to do with it? First of all, where to place it? Yeah, so we have an objective lens. It's the main tool actually we have as microscopists. We have a focal plane and we have a back focal plane, which is Fourier related to the focal plane. And typically we put our deformable mirrors or SLMs there, yeah, for two reasons. The first one is that every light ray that comes from the focal plane has to go through this nicely defined circular area. So especially if you have just a limited number of actuators on your SLM, it makes just total sense to put it there. You make most use of your SLM there. Secondly, it's also, I mean, the Fourier plane is pretty neat because some aberrations are global, like spherical aberrations, and the Fourier plane, whatever you apply here, right, will apply to every point in the focal plane the same way. So it's shift invariant. This is a nice property to have. Well, the Fourier plane for most objectives is inside the objective barrel. Of course, we can't put any bulky device there. So typically, you then just use a Kepler telescope to image your SLM onto that plane. And you have to make sure that you also match the right magnification such that your deformable mirror aperture matches the pupil size, right, which is 2 times f times NA of your objective lens. Right, so which path should be corrected, right? In short, because we always have excitation paths and detection paths in microscopes, in brief, it can be said, I mean, it should be that path that actually encodes spatial information about your object. Yeah. So for instance, in wide field microscopy, we know, okay, the floodlight, we don't need to correct the flooded light here. We just are interested in correcting the emitted light. So we put the SLM there. If you have weak unpolarized fluorescence, maybe you go for a deformable mirror. For the confocal, it's different, right? So also the excitation light is a spatially structured light. Structured illumination in principle needs to be corrected, maybe with the exception of rim, right? Because the speckle turns into another speckle. We don't care actually then. But in general, you would have to do that. And for multiphoton microscopes, the excitation path is the one that encodes the information. Yeah, the emission is irrelevant. We just want to collect it somehow in a bucket detector. And that's, I think, a pretty easy case because lasers are powerful. You have a lot of power overhead usually. There's also linear polarization there. So we can maybe use a pixelated liquid crystal spatial light modulator for that purpose. Right, so I want to spend 5 minutes on the refractive index mismatch because this is really, probably everybody who has ever used a microscope has actually faced or seen the effect of this spherical aberration mismatch. It can be pretty drastic. And what you see here on the bottom, that is actually an optically cleared mouse cochlea. But the clearing protocol actually leaves the cochlea at a very high refractive index. Yeah, and that if you go a millimeter deep inside, it's just nothing you can see. And this I think is really where AO can shine because well, it's easy to correct. Yeah, you can analytically calculate the aberration and then just display it, and you can have very high enhancements. So, well, why do we have this aberration actually? Why does the spherical refractive index mismatch introduce an aberration? This is because Snell's law is non-linear, right? So it means if you have a high NA, yeah, the marginal rays, the high-angled rays, they're kind of over-refracted in a way if you go into a higher refractive index medium, and therefore they focus at the deeper position inside your sample. Now those rays have high NA, so they build there at this position a tight focal spot. This is narrower than the actual area disk, right? But also have some rings. The paraxial rays, they focus way higher, yeah, and they have a low NA. So what you have there is this blob you see. Yeah? So if you go through your focal spot, you look at the single particle or a molecule, and you get this kind of asymmetric experience. If you look at your spot, then you know what you are facing. This is this spherical aberration from a refractive index mismatch. But it can be easily calculated. Yeah, there's not a lot of math now here, just a little bit. I want to deliver the intuition that is behind how this can be calculated, what I need to display on my deformable mirror to actually compensate for that effect. And it goes like this. So we start with a field in the focal plane, a perfect focal spot in a medium N1. Then we propagate back numerically on the computer. So that's basically done by a Fourier transform of the field. That brings me physically into the pupil plane. Yeah, this is the Fourier plane. Then I add a phase. Yeah, the phase is the phase of a lens. It's the Ewald sphere that you see here, D, the focus. And the radius of this sphere is the K vector of light in that medium, N1. Okay, I propagate back. That's done now. Then I insert the medium that I want to look into. Maybe it has another refractive index, N2. Okay, and then I simply propagate forward again. Yeah? And propagating forward means adding another lens with a different radius here because the K vector has changed, of course. In the medium N2, if the refractive index has increased, the K vector has increased. And that is actually my aberration. It's expressed in Fourier space. It's just the difference between two spheres. But my modulator is anyway in Fourier space. So what I need to display on my mirror is exactly this, and done. Yeah. Actually, it turns out I don't have even to correct the full aberration because that would not only make my focus nice and compact again, but also shift the focal spot back to a place where it would have been if there would not have been any medium N2. That's nothing that we actually need to do. In fact, actually, we want the spot to move downward because that gives us more working distance. And it actually also is a relief for the deformable mirror if we just subtract this defocus in the end to leave the spot where it is. So we leave the shifting effect of the refraction, but we cancel the effect of the aberration. And then you get this sombrero-like shape that you have probably seen a couple of times if you are into this AO. And you see actually this can be generalized quite neatly. Yeah, I mean, question, what do I have to apply if I want to look through a layered sample with many different refractive indices? It's actually very easy. In Fourier space, I just have to add spheres of different radii, and then I'm done. But that being said, yeah, AO is brilliant to correct this. But I have also to say there are objective lenses out there where you can set the refractive index, right? So this is possible to buy. There are also immersion lenses there which have a spherical front surface. Why is that good? Because all the rays are always at 90 degrees intersection with that front surface, and it doesn't matter what immersion medium I put there, there will never be an aberration because I don't have any refraction at that interface, right? So that's a special development. So if you have just a single refractive index you are facing with and you have the immersion, you can mix your immersion medium, maybe that might also be a way to go. Okay, so this is a known aberration. This is pretty neat to treat. Now, what about unknown aberrations? So there are, what can we do? One way is to do it like the astronomers. We can directly measure them, maybe with a Schack-Hartmann sensor, or there are also other sensors like pyramidal wavefront sensors out there. The concept of this is actually more than 100 years old to do it like this. The idea is that I have a lenslet array, and I have my wavefront that I send onto this lenslet array, and then I get spots at the back of a screen. In the past, it would be photographic film here. And then from the positions of those spots, I can directly figure out the slope of the wavefront, the local slope of the wavefront. And if I have the slope of the wavefront, I can basically, with some unknown constant, which is irrelevant, find out what the phase is. So the nice thing about Schack-Hartmann sensors is they work with white light, yeah? Which is pretty cool for a phase measurement instrument. But they also need, this is a little bit of the disadvantage, they need a spatial coherent source. Yeah? Why? It means what they are actually doing, what this Schack-Hartmann sensor is doing, it produces, so each lenslet is producing a little image on the camera chip of the object it's actually targeting. Yeah? Now, if I'm targeting a guide star, a little bead or something, I get maybe 100 images of beads, which are all a little bit shifted. No problem to find out where the centroids of these points are, and it will work. However, if I try to go really deep into the tissue, sometimes it's just speckle that I get back. Yeah? And then on the sensor, you will see kinds of like this. And these are situations where it's very difficult or maybe impossible to use these kind of wavefront sensing techniques. Nevertheless, it still works. Yeah? Again, by the Fragola group, light sheet work. Yeah? So it can be used for these kind of problems. Also in multiphoton imaging, it can be used. The mechanism there is that the focal spot itself, yeah, because I have a nonlinear effect, the focus itself is kind of a guide star, can be used to feed back, to be fed back into a Schack-Hartmann sensor, and that also works to some extent. So there's direct wavefront sensing. There's also a thing called indirect wavefront sensing, and I would probably say that this is the majority of systems in microscopy are working with this indirect wavefront sensing strategy. What's so special about it? We don't have a wavefront sensor anymore. Instead, what the name implies, right, indirect wavefront sensing, we try to figure out somehow by iterative measurements on my spatial light modulator what the aberration might be. Yeah? So I basically define, I always iteratively take images. I distill a scalar metric value from those images that kind of define the quality. Yeah, it could be the sharpness or the entropy or the intensity, the total intensity in the image for nonlinear imaging. And then I try to climb up that ladder, right? And the higher the metric gets, the better hopefully gets also my image. And there's like a plethora of different implementations that have been done. There's modal wavefront sensing, zonal wavefront sensing, you can display Hadamard patterns, genetic algorithms, and so on. But if you look at companies that are out there providing AO for us, basically, and there are not many, maybe that list is not really complete, right? If you look at what they are doing, so those are companies that really provide you the whole bundle, not just selling you spatial light modulators. If you look what they are working with, how do they measure aberrations? Most of them are using a thing called 3N algorithm, yeah? Gradient search, which is basically the same 3N algorithm and so on. So I want to say a little bit about this 3N approach. Yeah? It seems to be a very efficient way to measure aberrations. How does that work? Well, first of all, we make life simple. We make a series expression in my pupil. The phase I have there is expressed as a series of orthogonal modes. Yeah, Zernike modes is one example, but it could be eigenmodes of your mirror. Important is they are orthogonal. Then secondly, I define a metric value, as I just said, yeah, maybe the sharpness or the intensity. And then I just make test measurements, and I am assuming, since I can assume orthogonality, yeah, that each of those modes has an independent effect on that metric. Yeah? So it means that if I, let's say, measure and correct mode 6, I don't want to degrade mode 5 again. If that's the case, I can actually find out the aberration content of each mode in 3 measurements, yeah, like this. This is done for coma here. Maybe I do an image without displaying anything on my SLM. I measure metric value M0. I display a little bit of negative coma. I measure a lower metric value. I put a plus bias coma on there. I measure a higher value of coma. And now, under the assumption that the aberration is not too strong, and this is a very important assumption because I will be coming back to this assumption, I can kind of say, okay, I'm very close to the optimum already, fit something like a parabola through those 3 points, and then find out what the aberration content of coma is in my system. And then I go to the next mode. Yeah? Hence the name 3n. I mean, theoretically, n plus 1 are sufficient, but nobody does it like this, right? So 3n measurements is enough. I was actually also asked to bring my setup along. I didn't know anything about me4bio and the concept here, so I was a little bit puzzled. So I couldn't bring my setup, unfortunately, but I thought maybe I do the next best thing. I just make a movie of my desktop when I'm running this 3n algorithm in my lab. Maybe you get a little bit of a feeling how this looks, how long it takes, and so on. So here I'm looking, so this is my homemade LabVIEW screen. What you see here is a human brain organoid, yeah, 2-photon microscopy, by the way, GFB expressing neurons. Yeah, we see here on the left, you see a wide field image. Basically, you see nothing because it looks like a cotton ball. It's like a really scattering thing. This is a 2-photon image, maybe at the depth of 100 to 200 micron. And on the top, you see the panel of the deformable mirror. On the right, top right, you see the shape it has at the moment. And at the moment, it just takes a shape that flattens out its own surface, like an offset correction shape. Okay, so now. So what I'm doing here, I'm selecting a cell that I want to have measured. Then I click on 3n. Then you see this window popping up. You see the mirror sweeping through the modes. So you see maybe this white parabola fits. And then that was correct in 33 modes. And then I do again a scan, and then it has been improved, right? Okay. This is exactly this result that I've just taken. It's unprocessed, so no denoising or deconvolution before I did AO and afterwards, okay? So they're both not the best images. I have to say those are fixed organoids, already like 2 months old, slowly degrading, and GFB is now tending to be everywhere. But I think you can see that the AO image is better, right? You see the dendrites here, which you don't see here in the not corrected image. Okay, you can make this work pretty robustly, actually, but there are a few pitfalls that I stumbled over when I was a postdoc. And I want just to speak about, well, orthogonality is such a thing, right? But what I found even more important is to work with shift-free modes. Yeah, what do I mean by that? If you don't do that correctly, it can totally fail. Or even worse, it sometimes works, sometimes not, and you don't know why. So what's the problem? I just said before these test modes, so this 3n algorithm that applies test modes. Yeah? So I add a little bit of coma, a little bit of spherical aberration, and so on. Now, if you have an underfilled pupil, and most scanning systems have underfilled pupils, especially nonlinear ones, yeah? Then, if I display, for instance, primary spherical aberration, like this, looks like a sombrero, as I have showed you before, the beam actually only crosses through the center. Yeah? And the beam actually mostly sees defocus, not a primary spherical aberration, and therefore the beam will shift whilst I do my test. And if I'm really unfortunately, it could be that my beam actually gets worse, the focus elongates, but it shifts into a fluorescent layer. And then my signal goes up. Yeah, I think you understand the problem. It means that I actually measure an aberration which is not there because my algorithm is full. The signal, the matrix goes up when I aberrate my system. And then I really make it worse. The next mode will not be properly found. I come to the next mode. There's also a crosstalk between tip tilt, so the shift modes, and all Zernike modes that have the same azimuthal symmetry, right? So coma will also shift sideways, same problem, and then it will just collapse, right? It will not work. Fortunately, you can undo those shifts, and this has been nicely described by Tile and Booth. There's a paper, not very prominently published actually, 2011. If you are facing this problem, make sure just to look into here. Everything is described here, yeah? And it's actually very easy to do. You can just measure the shifts you have in your system, maybe with the beat. Yeah, we just apply every mode in turn. You measure the shifts they do. You get a shift matrix like we have here, and then you can kind of, using a linear algebra approach, just get rid of those shifts and work with the new basis. I'm also mentioning this because we recently had a problem or a work where this was very essential to do. And what we did here was we were retrofitting a commercial Raman, confocal Raman system with AO. So what we wanted to do, we wanted to have minimal engineering. So not to undo kind of the whole system, build in some 4F system and so on, and need an optical table. The only thing we need to do here, because we use this optofluidic element, which we could just attach between the nose piece and the objective lens, we just needed to place it there, essentially, right? That was pretty easy to do. It even has a Thorlabs thread already. Okay, but we had to take into account these shift modes because the laser beam was extremely narrow, yeah? Probably because they wanted to make it fit also for small pupil objective lenses, but then if you take larger ones, you have a very largely underfilled pupil. And then we got extreme shifts. What you see here on the left, this is like a frontal section through a Raman image through a polystyrene bead. In the middle, you see what happens if I apply Zernike spherical. You see the image goes up dramatically. And on the right, you see an image when I apply the shift-free version of spherical aberration, and you see it stays in place. I can nicely use my algorithm. So what we did here, we applied this 3N algorithm, but we could also do another neat thing. So because it's Raman, yeah, has a lot of information, we did something that we call spectral guide star sensing. So we have the spectrum. We wanted to look into brain, yeah. But sometimes you find erythrocytes, red blood cells, that have a very strong peak at 1600 wave numbers. So we're using that peak to inform our system to increase the signal by a factor of 2 in this case, and that gives us also then the opportunity to measure spectra from the surrounding tissue at also an increased signal level. So the main motivation for this work, I think, for Raman is not really that you get sharper images. Of course, you also get sharper images, but it's more a saving in time. Yeah. I mean, there are super fast Raman systems around there. Yesterday, I've seen one in action. It's actually also here at me4bio. It's worth visiting. So Hilton showed me how it works, or his team, actually. But if you don't have a fast Raman system, those scans can take hours, yeah, depending on how weak your signal is. And if you can then increase your signal by a factor of 2 or maybe even more, you just save 50% in time. And I think if that's a routine operation, it's really something worth following up. Okay, so what's beyond 3N? Most use 3N, but there are microscope modalities where you actually don't need to do any test measurements. Yeah? And one such a thing or modality is single molecule imaging and tracking. Yeah? Why does that work? We don't need any test measurement, still we find out aberrations. There's a couple of works have been published in the recent years, including from my own team. And the idea there is simply do deconvolution in a way, right? Because, I mean, in deconvolution, usually you know your point spread function in ideal case, but you don't know your object, so you deconvolve it. In SMLM, it's the other way around. You kind of know your object, it's single molecules, but you don't know the PSF. But convolution is commutative, so you can apply it to find out your aberrations. And then you can do either digital AO, yeah, that gives you then a higher success rate when fitting your molecules. It doesn't increase the precision though, right? That's an important difference between digital and physical AO. Or you can feed back the found shape to a light modulator, then you do physical AO, you increase the physical information in your raw data and therefore get a higher precision. So that was the tutorial part, so to speak. I would now like to address another topic, which sometimes is referred to scatter compensation. Yeah, I mean, scatter compensation is basically the same as adaptive optics. There's no physical difference. Yeah, the origin is just refractive index inhomogeneities. But when your images start looking like this, you might call it scatter compensation, right? For me, the difference is that in this kind of regime, this 3N algorithm just doesn't work anymore. Yeah, it will not work anymore. Still, it's possible to do it, right? Those are 4 micron beads. Okay, they have a lot of fluorescence, but they're hidden under 0.5 millimeter chicken breast tissue. It's really, really a strong imaging problem. Yeah? If you see nothing, even with the 2 photon, if you try to look through it. Why does the 3N fail? I mentioned before, there is one important assumption. The assumption is that I have small aberrations, yeah? Because otherwise, I'm not close enough to the local optimum in my, or to the global optimum in my optimization space. I'm somewhere in my n-dimensional optimization space. And then the optimization problem is similar to those optimization problems you solve on a computer. It's really hard to find the global optimum. Luckily, there are tools. Yeah, all these Giphy videos don't work. It's a bummer, actually. But there are methods that make it work. Yeah, and those came up around the year 2007. This range, and the most important one, or the most famous one, I have to say, is the one by Velikop and Mosk, who showed that by controlling a limited number of modes, yeah, pixels on a spatial light modulator, basically, I can still see something through a totally diffuse medium. And I refer to these methods as interferometric methods. So why? Because they actually do interferometry. Yeah? How they work is, at least in this demo paper here, what they did, they just moved every single pixel on a pixelated SLM in time. And therefore, in the fluorescence target, or in the focal space, right, if you have a bead, right, a fluorescence bead, what you do here is phase stepping interferometry. So I shift the phase of this single pixel, and I will get a sinusoidal intensity excursion that I can measure, right? Okay, it's interferometry. We know we need 3 measurements to find out what the phase is, the amplitude, and the offset, right? 3 unknowns, 3 measurements. Now, why does that solve our problem? Because the solution must be within the interval 0 to pi. The optimum must be somewhere within this interval. There's no way that I can get lost in my optimization space. I think this is the crucial difference to the other model-based waveform sensings, that this actually works also for the scattering environments. However, there are 2 problems. The first is, okay, I cannot really use a sharpness metric because there is no sharpness. I just see a blur. And secondly, it does not really work with linear fluorescence unless I really have a guide star. Why is that? Because those 2 cases, extremely scattered case and the scatter compensated case, give rise to the same amount of fluorescence photons in linear fluorescence, right? Because it's just the integral over the total intensity in the target plane. If there would be, let's say, a 2D fluorescent C here, yeah. So my metric would always stay constant, no matter what I do with my SLM. It's not a useful information source for my aberrations. It doesn't work. However, for nonlinear microscopy, then it will work, because a focused spot just gives higher signal than the same intensity spread out over the full 2D fluorescent C, right? And the second problem with this original Velikop approach is that, let's say, I want to measure 1,000 pixels. Okay, if I just move 1 pixel at the time, I just have a contrast problem, yeah? I just control 0.1% of my total intensity, and I try to measure like an interference fringe, and this is just not really practical. It just doesn't work in practice. So we have a couple of years, or maybe it's now 4 years ago, published a method we call DASH, Direct Adaptive Scattering Compensation Holography, which kind of at least solves problem 2. And it's heavily influenced or inspired by another technique called F-sharp that was published by Ben Yudkowitz in 2017. Yeah? So F-sharp has its really good properties. It's very good if you have a slow light modulator, but it's a little bit more complex. Dash is more simpler to use, as I will show you now. And it has a faster convergence rate. So rather than testing single pixels, what we are doing here is we are testing plane waves. Yeah? So we're creating plane waves directly by the light modulator via displaying a phase hologram, hence the name Dash, right? Holography. And we do that by just adding on a computer the fields that we want to produce. For you see here in blue, 80% light should go into a field called C. That is my reference wave. At the beginning, it is just a plane wave because I don't know anything about my aberrations. And I put 20% of the light into a mode, one single plane wave mode. Yeah? And we would typically start with the ones that have the smallest angles. Yeah? So we start in K-space in the center, and then we spiral out in K-space and increase and continuously increase the angles of my plane waves. And then it works basically similarly. Yeah? We choose a test beam, we phase step it, we measure the phase, the relative phase between my plane wave and my reference, and the amplitude. And then we immediately update the reference. I think this is an important step also to ensure fast convergence. We immediately update the reference beam with that information by adding the tested plane wave mode at its optimal phase. And we just go back and choose the next plane wave. Okay, that should be a video. I didn't test it properly. I'm sorry how that actually works in a simulation. But anyway, this is real now. I would like to show you also a Dash experiment. This would be our control experiment, just to check if we've done everything right with the electronics and our setup. And our test experiment consists of having beads or just a dye layer under a cover slip and a layer of Scotch magic tape. Yeah? Magic tape, it's the white one where you can write on it, it's scattering. And you see here in the two-photon image, it's really blurry. You cannot really see those beads anymore. Yeah. Okay, so let's run this. So what I do, I have to mark where I park my focus to do the measurement. This would be here in the center of the green square. Then I click on Run Dash. Then you see another window popping up, and you see, now it's already done, right? You see how the pattern of the mirror evolves. And afterwards, this is the final shape of the mirror. Here I corrected 300 spatial modes. And then after the scan, again, you see that you can actually clearly see those beads. Right, so this is the result of exactly this measurement. These are those 3 beads before and after correcting using this scatter compensation algorithm. And here we got an enhancement factor of 4 in this case. Usually for this one layer, we get an enhancement of 4 to 5. Then we know, okay, it works. Luckily, that doesn't only work for strongly fluorescent beads, it also works for fluorescent proteins. So this is an example we got from GFP expressing microglia in mouse brain tissue. Actually, I don't know how much time I have. You're giving me a warning, right? 5 minutes? 5 minutes. Yeah, here we have similar enhancements. This is 200 micron deep in mouse brain. So you've seen this was maybe 4 seconds, yeah, to go 3 iterations. We are not pushing it. At the moment, we are measuring the PMT signal, going to the PC, calculating the new pattern in LabVIEW, sending it to the PCI Express card of our mirror, and then it goes to the mirror. So we are measuring actually only at 1 kilohertz. Yeah? The mirror can do 20 kilohertz, so I'm not saying we can do it 20 times faster, but I think we can do it 10 times faster. Yeah? So in principle, we could go down to 1 second for correcting 500 spatial modes. This is when you really push it. Yeah, I think what I wanted to say, however, it's not super immune to background, right? Yeah. So it works best if you have sparse labeling. Yeah. So for these organoids, you might have been wondering, why don't I show a Dash video of these organoids, right? It's just not as fast there because we have a lot of background there. But it does still work. Yeah, I will show you an image there. But one of the nice properties of Dash actually is that it's alignment-free. Why it's alignment-free? It's not fully alignment-free. You need to make sure that the laser that bounces off your spatial light modulator goes into the objective lens. Yeah, you need to make that sure of that. But it's not as with the formable mirrors where you have to really be precise with the alignment by 1/10 of a millimeter. Yeah, You only need to make sure that the beam somehow goes into your objective lens, because the pattern bases are plane waves. And plane waves are plane waves, right? The pattern will just build up wherever it's best for the image. I think this is a really nice practical property of the algorithm. But it needs a very fast diffractive element. Yeah? This is a downside, yeah, big downside. And those used to be super expensive in the past. Yeah, you can pay a six-figure number for those mirrors. Luckily, the things are going to change. Yeah, maybe some of you already noticed there is Texas Instruments since about 5 years developing a new type of light modulators based on their light projection technology. You probably know these DMD mirrors that are these digital flip mirror arrays that are working in projectors. Based on the same platform, they are now releasing piston mirror arrays. Yeah, exactly what we need, exactly what DASH needs actually. These are mirrors that move up and down, and you have a megapixel of them, and they are really fast. So at the moment they run at 1.4 kilohertz, only at 16 different levels, but it doesn't really matter, to be honest. And a month ago we had managed to do the first test measurement running DASH on one of those mirrors. So that is done the blue curve. You see here the signal enhancement versus time. The blue one is correcting 400 modes with this MEMS mirror. That was done in maybe 2 or 3 seconds. And the red curve was done with a fast liquid crystal light modulator that was running at 300 hertz, which is already pretty fast for a liquid crystal device. 1 or 2 minutes or so for challenges, right? Because it's not, the world is not beautiful. Not everything is beautiful. What are the biggest challenges now for this? One challenge is, well, what is it good for, right? Because it turns out, so this is showing again organoids, yeah? You see on the left, no AO, in the middle, that was 3N using 33 modes. You see that it's actually a little bit sharper, right? And that was using DASH, right? With 200 modes. You see the signal goes up much more, but the validity of this correction is very narrow. The more spatial modes I include, the narrower gets the so-called isoplanetic patch. Yeah? So the dream of like getting a square millimeter of a brain organoid in a depth of a millimeter is just, we are not yet there by far, I would say, right? Because this is just giving me, you see at the scale-wise 10 micron, a 10 micron field. Another example from extracted lung mouse tissue with GFP expressing endothelial cells. If I go 30 micron deep, everything is fine. I get some enhancement. If I go 75 micron deep, I get like a huge enhancement, but a patch that is only 3 micron in size. Okay. So what you can do is you can just move your SLM into a sample conjugate field. That is something you can do. Yeah, we did that. And there you can simultaneously correct multiple points. That is something you can do, right? So here this is showing microglia cells in fixed mouse brain tissue where we corrected 5 at the same time. So DASH has to run sequentially because I only have a PMT. You can't discriminate with the signals coming from these different points, so you have to run the optimizations in sequence. But all are displayed at the same time at my spatial light modulator, so all are at the same time also corrected. So I could imagine having an AOM moving around my beams very quickly between the corrected points. And this is also not the limit, right? I guess you can upscale that maybe to 50 spots, maybe 100. The last thing I want to say is speed, right? Of course, in live tissue, you have decorrelation with time. This also depends on the number of modes you include. Some are measuring over hours, yeah, with the corrected field in live mouse brain. We tested it at least not in vivo. Yeah, I have no experience with in vivo, but these are live mouse brain tissue slices. They can be kept alive in a tissue culture. And we, again, the GFB expressing cells, we did corrections, measured how long are the correction patterns valid, and we measured around 5 minutes on average, which is not a bad result, right? If you imagine you do a correction in a second or a few seconds, if you then have 5 minutes, it's not too disappointing, I would say, or disencouraging. So if you want to play around with the DASH algorithm, yeah, we just published together with Kibom Nam and Chang-Hoon Park from UNIST an updated version together with some Python examples on GitHub. Feel free to download and try them out. And with that, I think it's just left to thank you for your kind attention and the team at our institute and the funding. Thank you very much.
insightful talk. So I'm a bit curious. You want to avoid focus shift inadvertently, right? Because you can accidentally get into this region that's fluorescent and your algorithm goes astray. Now, I didn't really get how you did that. I mean, as you seem to mention bead measurements, but how do I do that in a sample that I don't really know? So I didn't really understand how that works.
How the correction actually works, yeah.
Well, how do you ensure that you don't get this unwanted focus shift happening?
Yeah, I mean, there is one effect. That's a good question, actually, because there is limited control of where the focus actually will form once it's corrected. Because you actually also need a kind of a guide star, even if you do multiphoton imaging. But what happens actually is, in the very first measurements, you have a speckle pattern on your sample. And how it works is that the brightest speckle simply wins. So the first few measurements are pretty uncertain. Yeah, you don't really get nice sinusoidal signal excursions. It is just the speckles are fighting. And then very quickly, it was actually a GIF that I was unfortunately didn't work, but then you see very quickly one speckle wins, and that is then all the energy will go there. But I don't know where that speckle will be. It's a combination of where is my fluorophore density distribution, and how does my scatter pattern look, basically, how does my speckle pattern look, yeah. I mean, once the speckle is there, I can scan, and I will get an undistorted image. But there is an unpredictable slight XYZ shift that I cannot really control. I mean, the only thing I can do in the end, if I detect a large defocus component in my phase pattern, I could try to get rid of that, for instance. But where the focus exactly will form, I don't really know. Yeah? But I don't need a guide star, right? Yeah.
Okay.
Any other questions? Don't be shy. So, yeah, okay.
Thank you very much for the talk. For the application in the Raman microscope, so you have this very nice adaptive lens. You just seems to be very simple to use. You just put it at the back of the objective.
Yes.
So it's not really conjugated to the backfocal plane, right?
That is,
can you comment on that?
Yes, that is totally correct. Yeah, it's not at all in the backfocal plane. But for this type of microscope, since it's a stage scanning system, it just is totally relevant, right? Because actually the beam is always just going straight into my objective lens, and it doesn't matter it doesn't really matter at all where the light modulator it is. If I would have a scanning system, of course it would be a different thing. But yeah, for that system, I think it is not an issue.